Hi everyone, I am Sutapa present in front of you again. Welcome to my channel, Sutapa's Biology. My today's topic is cell division. I am continuing the last topic, that is cell division 1 we have completed, and now today's cell division 2. Another thing, hope you all are well, you all are staying safe and happy. I am coming to my today's topic, that's cell division 2. In my last video, I have told you about the interphase of the mitosis. Now today I am giving description of the divisional phases, and in the divisional phases about the mitosis you have already started in your lower classes. Class 8 to 9, 10 I think you have started in those classes. So I am giving the short idea about it, and I shall give the illustrated view about the meiotic prophase 1. Okay, so now I am starting the divisional phase, that is the M phase. Divisional phase of mitosis, M phase of mitosis. I think you know about it, that the M phase has 4 subphases, that is in karyokinesis. Broadly it can be divided into karyokinesis and cytokinesis. Karyokinesis, and cytokinesis. Now karyon means nucleus, so karyokinesis means division of the nucleus. It's the division of the nucleus, and it's again divided into many subphases. लास्ट वीडियो आप लोग देखे हैं ना, उसमें हम जो डिस्क्राइब किए हैं उसको कुछ कंटिन्यूएशन इस वीडियो में है. सो इसीलिए पूछ रही हूँ मैं, जो आप लोग जरूर लास्ट वीडियो में वॉच किए हैं तो आप लोगों को सुविधा में आएगा ये वाला. That is the division of nucleus, and this is the division of cytoplasm. Now karyokinesis has many subphases. Number 1, prophase. Number 2, metaphase. Number 3, anaphase, and number 4, telophase. So these are the subphases of karyokinesis. Now in cytokinesis there are no subphases like that. Only in cytokinesis we shall know about the different procedure which is followed in plant and animal. Plant में and animal में वो अलग अलग होता है cytokinesis. वो इसके बारे में हम लोग डिस्कशन करेंगे इसमें. Okay, so first I am discussing about the karyokinesis, the subphases of the karyokinesis, that is the prophase 1. In interphase you have studied, I think so you have studied as you have followed my last video. So there I have told you that the nucleus, all the nucleus, they remain in the lumped condition, that is like the woolen tuft, और मतलब ऊल के जो ये होता है ना एक साथ रहने से जो क्लम्प हो जाता है ऐसा लगता है. मतलब कोई different chromosomes are not observed. No chromatin fibers, no chromosomes are observed. Only the tuft of wool-like lump is observed. That's in interphase. Now, in the first stage of the first subphase, that is prophase 1, only prophase. the chromosomes are taking shape. That is, chromatin fibers, they are making the chromosome like threads. Those are separated from each other in this manner. Not very prominent, as I am drawing, it's not so prominent, but like this. That is not the lump. It's different, all the chromosomes are different here. Within the nucleus. But with this, the nuclear envelope and the nucleolus both are gradually disappearing. So nuclear envelope here are drawn very prominent, but it will not be so prominent. It will be gradually faded. So nuclear envelope is gradually disappearing with the nucleolus as well as the nucleolus. So I am giving some this type of appearance here. The nuclear membrane is disappearing. As well as the nucleolus is also disappearing. Suppose this is the disappearing nucleolus. Now this is about the prophase, and after that, after prophase, in metaphase, there is the prominent alignment of the chromosome. Condensation of the chromosome is completed. Here started, and here in metaphase, the chromosomes are mostly prominent, and that's why we can study the metaphase plate, and from the equatorial plate of the metaphase, we can count the chromosome number as well as the length of the chromosome. That is karyotypic study. What is karyotype? Karyotype is the study of the chromosome or counting of the chromosome, and also the how many chromosomes are the long, which are short, etc. All this description we can get by karyotype study. And another is after, okay, I am giving you this terminology, that is karyotype and ideogram. Karyotype is the study of chromosome in the metaphase plate. In metaphase plate, and ideogram is the pictorial representation of that. Ideogram, that is the pictorial representation of karyotype. Of the karyotype. So in metaphase plate, metaphase, we can see that the complete condensation is completed, and the chromosomes are mostly prominent, and all are arranged on the spindle fiber. And spindle fiber formation is also completed naturally. Spindle fiber is formed from the centriole in case of animal cell, and in plant cell they are formed from microtubule, as plant cell do not have the centriole. In animal cell, centrioles are very prominent with asters. And here we can see that the chromosomes are moving in this manner. Further due to further contraction of the spindle fibers from the next one, that is from anaphase, we can see the different configuration. But in metaphase, we can see all the chromosomes are arranged in this way on the equatorial plate, and this is known as metaphase plate, wherefrom we can study the chromosome prominently. In competitive or in board also, you will get the question in this manner. Name the subphase where we can count the chromosome as well as we can observe the chromosome very prominently. That's the metaphase. So after metaphase, that's the anaphase. I am rubbing this one. Anaphase is prominent with the Anaphysic movement, and here in the metaphase plate we are observing that the chromosomes are arranged in this manner, but these two are the chromatid, not the chromosome. They are the part of the same chromosome, that is, these are the chromatid. Now in anaphase we can see that the centromere is completely separating the two portions, that is centromere has two kinetochores. The two disc-like protein structure known as kinetochores present in the centromere, that is, if we draw the centromere in this manner, this is the centromeric region, these are the two kinetochores which are disc-like structure, kinetochore, present in all chromosome. Now these kinetochores, they will be separated in anaphase, and naturally the chromosome will be split to form two chromatids. Now one chromatid will go to one pole and another in another pole, and during this anaphasic movement they take different shapes according to the position of the centromere. Now which type of shapes they are taking, I am discussing about that. So in anaphase they have the movement which is known as anaphase. The chromosome show the anaphasic movement. What is anaphasic movement? I am explaining. Like I have told you that centromere of the chromosome, suppose this is the chromosome, it has the centromere in this manner, and each chromosome has two chromatids, and for one chromatid one centromere. Not centromere, for one chromatid one kinetochore. So in anaphasic movement we can see that the chromosomes are split to form two chromatids which are moving towards the opposite pole with their own centromere, and then they will form their own chromosome also. Now according to the position of the centromere in the chromosome, the different configurations will be formed during the anaphasic movement. क्या क्या है वो? Like that, in case of metacentric जो है उसमें क्या है, just at the midpoint the centromere is present. So both the arms are equal in length. दो chromosomal arm एकदम सेम साइज का होता है, तो इसीलिए during the anaphasic movement they will take the configuration of the V, the English alphabet V, in case of metacentric chromosome. Now next one is submetacentric. During submetacentric they will take the configuration of L. Why? क्योंकि उसमें दो आर्म इक्वल नहीं है, एक थोड़ा बड़ा है और एक छोटा है, तो इसमें क्या होगा, एक छोटा और एक बड़ा, तो ये जब पोल की तरफ मूवमेंट करेंगे तब ये एल शेप लेंगे, एल. So this is submetacentric, and another type is that is acrocentric. And in case of acrocentric, one arm is very small in comparison to other. एक बहुत छोटा सा आर्म है और एक उसके हिसाब से बहुत बड़ा है. So it will take the configuration of J. It will take the configuration of J. One arm is like this and another is like this. So during anaphasic movement, it will take the shape of the J. J. And this is acrocentric, and last one is telocentric. Telocentric has only one arm and centromere at the terminal region. So in case of telocentric, they will form the configuration of I. One arm is very big and there is no other arm. So it will take the configuration of I. This is telocentric. So here from we are getting 4 types due on the basis of the position of the centromere. Centromere की position के हिसाब से ये 4 kind का होता है, जो type का होता है, जैसा metacentric B, submetacentric L, acrocentric J and telocentric I. Now after the completion of the anaphasic movement, the chromosomes will be at the polar region and they are equal in number. They are separated exactly equally. Now and after completion of the anaphase, the anaphase में क्या है? Anaphase में nuclear membrane थोड़ा सा भी नहीं दिखाई देगा, completely disappeared, as well as the nucleolus. Now from the metaphase also, in metaphase, late metaphase also, we can't get the nucleolus and nuclear membrane at all. Now next is telophase. Telophase is the re-establishment of the interphase, like that. Re-establishment of the interphase. Telophase का जो diagram है वो just like the interphase. Only interphase में जो एक दिखाई हम लोग देखे थे, एक जो था ये 2 बन गया, यही just difference है. So in telocentric, the nuclear membrane will appear. Telophase. In telophase, nuclear membrane will appear. Chromosome will form the lump again, as well as the nucleolus will appear in both the cell. So now the cell has 2 nuclei and only 1 nuclear envelope. This is the telophase. Now after telophase, there will be the cytokinesis. So telophase is the last subphase of the divisional phase, and after that cytokinesis will take place. And in cytokinesis is completely different in plant cell and animal cell. In animal plant cell, they form the cell plate by formation of the phragmoplast. In my cell wall video of the cell wall, I have described all about the phragmoplast, the structure of the phragmoplast, and phragmoplast is formed from the Golgi bodies and is sometimes ear, and as well as different other materials which are required for the formation of the cell wall. So phragmoplast is present or will be formed in the plant cell, which will separate to. So now in cytokinesis we can see 2 type of in plant cell it will be like this, and in animal cell it will be like this. Now in plant cell there is the formation of the cell plate from phragmoplast. In between the two, that is in between these two there is the formation of the cell plate in plant cell. But in animal cell there is the formation of notch. There is the formation of notch which will divide the two cell differently. Phragmoplast, this is phragmoplast. This will form the cell plate, and this is the notch or furrow. This furrow will deepen and they will touch each other, and then ultimately they will form two different cells. So this another thing is there, that the significance of the mitosis. Significance of the mitosis are mainly that the mitosis make the growth of the organism. From one cell unicellular condition, that is from zygote, where now the complete person, adult person or like that, that's due to the mitosis. So it has a great role for the growth of the organism. And number two, in case of any cut and done, in case of plant and animal everywhere, the mitosis helps to repair it. Okay, मतलब हम लोग का हाथ काट गया, मतलब वो क्या कटा हुआ ही रह जाएगा? वो नहीं, सेल डिविजन होके वो 2 दिन बाद ठीक हो जाएगा. It will be repaired by mitosis. Another thing is that the mitosis helps in the different help in the different organism to produce their own kind. Like that, in asexual reproduction, we can get the organism can make their own kind by the cell division mitosis. So this is about the mitosis, and my next video you will observe about the meiosis. After that, bye to you.